DMT stands for N N-Dimethyltryptamine, and it's a powerful psychedelic compound found all throughout nature, including inside of you in the form of endogenous DMT. DMT has been found in human cerebrospinal fluid and trace amounts of it in human blood and urine. A precursor to DMT synthesis has been found in the lungs, with some suggesting the lungs to be the primary source of endogenous DMT. It's been speculated for years that the human pineal gland produces DMT, but there's very little evidence to show that at this time. Exogenous DMT can be synthesized in a laboratory setting or extracted from plants throughout nature. It's typically consumed by vaporization, but it can also be smoked, injected intravenously, injected intramuscularly, or it can even be swallowed orally as long as it's done so with the presence of an MAO inhibitor. Dr. Timmerman and his crew decided to go with the intravenous route because if you're going to give someone a powerful psychedelic, strap an electrode net to their head, and then shove them in an fMRI machine, I mean, that's going to be the way to do it. They began taking the simultaneous EEG-fMRI measurements 8 minutes before the DMT bolus was delivered and then stopped 20 minutes after the delivery. Now you may be thinking, that's a pretty short amount of time. I mean, that's basically a half an hour, but that's one of the most amazing things about DMT is that despite being a very powerful psychedelic, the trip is extremely short, usually lasting around 10 to 15 minutes. Using what's known as the fMRI bold signal, it's possible to determine blood flow and oxygenation to various brain regions, suggesting the connectivity of those regions. With the fMRI, they were able to determine how metabolically active the PTO was at rest, along with multiple other networks in the brain. When they compared the resting activity of these networks to the activity during the peak of the trip, they found something really fascinating. The integrity of the established networks, like the PTO, became less cohesive, and the boundaries of separate networks throughout the brain actually became less segregated. So that means the neurological lines were blurred in regards to their connectivity. So put another way, regions of the brain that started working together that normally don't, and regions stopped working that normally do. Now there's a few notable exceptions to this. The salience, frontoparietal, and default mode networks were actually shown to have an increase in functional connectivity. These networks are found all over the brain, so it's not really easy to show you exactly where they are, but it's important to note that they're responsible for high-level cognitive functions like attention, decision-making, and what's known as self-referential processing, which is essentially processing information about yourself. This suggests that DMT has the ability to not only dysregulate established networks, but open up connectivity between networks, all while increasing connectivity in regions associated with attention, decision-making, and information processing that's relevant to oneself. It's super cool and also just the tip of the iceberg. I've been using this term transmodal quite a bit, so it's probably best that I at least explain what a modality is. In neuroscience, a modality is in reference to a stimulus. So for example, vision is one modality, as is hearing, touch, scent, and taste. If a brain region only processes one modality, it's what we call unimodal. A good example of this would be the visual cortex, which I'm circling here. And this is what processes visual stimuli. If a brain region processes multiple modalities, it would then be called multimodal. For example, there are parietal and temporal regions that fuse visual, auditory, and somatosensory information. Transmodal regions process information in an abstract way, independent of a specific modality. So an example of a transmodal region would be the cingulate cortices. So this is the anterior cingulate, and then we have the posterior cingulate. And they belong to the frontal lobe or frontal cortex. And these regions receive information from unimodal regions and multimodal regions, and it integrates them into higher order representations like concepts, goals, and rules. This information handoff is a neurological hierarchy. Information is first processed at the unimodal and multimodal levels and then given to the transmodal levels for high-level processing. Dr. Timmerman and his crew discovered that DMT essentially compresses this hierarchy, making them less distinct. Basically, unimodal, multimodal, and transmodal regions began to look functionally similar. And this doesn't necessarily mean that unimodal regions began processing abstract information, since that would require them to start processing information in more complex ways than they're likely capable of doing, but it could suggest that transmodal regions began processing information at a more grounded level. To my understanding, it's just not possible at this time to determine exactly what this means without speculating, but it does seem to be important to that psychedelic experience and could provide some real insight into the nature of consciousness and cognition as a whole, because Dr. Timmerman and crew also discovered that the regions most affected by DMT happen to have the highest density of serotonin 5-HT2A receptors. Serotonin belongs to the class of monoamine neurotransmitters, along with dopamine, norepinephrine, and epinephrine. It's derived from the amino acid tryptophan and is produced in both the brain and digestive tract. Serotonin binds to a group of receptors known as the 5-HT receptors. These are G-protein coupled receptors and ligand-gated ion channels found all throughout the nervous system. There are many subtypes of 5-HT receptors, and the 5-HT2A receptor is specifically significant to us because besides binding to serotonin, it also binds to numerous psychedelics, including DMT. LSD, psilocybin, psilocin, and mescaline are also known to bind to 5-HT2A receptors, so we know this receptor plays a massive role in the psychedelic experience. Previous positron emission tomography, or PET imaging studies, have mapped the distribution of 5-HT2A receptors in the human brain, finding the highest density of 2A receptors are in higher order cortices, such as the posterior cingulate cortex, or if I flip this over, we can see the temporal cortex. Dr. Timmerman found that these same regions were most affected by DMT in terms of their increased connectivity. And this lines up with previous data and provides further evidence that the 5-HT2A receptor underlies DMT's effect on the brain and consciousness. Dialing into the effects that psychedelics have on these receptors is going to be very beneficial to our overall understanding in the long run.